Turn this pile of golden foil into a Japanese polished foil ball. And then ask me if it works out. I'm going to put it in my vacuum cleaner and see what happens. Okay, first we'll take my golf foil ball. Okay, now let's start timer this tool.
Good morning, everybody. How's everyone doing today? It is 8 o'clock We'll just chill here for just another moment or so, make sure everybody's finding the right location. How was everybody's Christmas break, New Year's break? Everybody stay local?
Yeah, I stayed local, didn't go anywhere.
I went to Indiana.
Indiana. Excellent. That's where you're from?
Yep, and 2 of my sisters had babies within the last 4 months, so I finally got to meet them, so that was fun.
Indeed. Congratulations to the fam.
Thanks.
Well, it's good to be back together, everyone. I see that our numbers are tending to plateau. We're right around 30 folks, 28 in the group. Is that what we anticipate? Maybe so. Let me begin a recording in case we need this later. Okay, let's go ahead and begin. Thank you, everybody, for being here. And apologies for yesterday not working out. Evidently, over the break, they've purged all of our personal Zoom accounts on the part of faculty and have a generalized one set up, but I guess no one really had to know that. We're good to go and set. Yesterday would have been just a short interaction. So if you've seen that email, thanks for walking through those general ideas. I posted this document both on Canvas, which you could see over here, heads each of the modules that I'll be responsible for, and then also sent it through that email as part of that email yesterday. So yesterday, very quickly, we would have generally introduced the block, at least the parts that I'm responsible for, and kind of preempted any confusion maybe as far as the logistics or how we'll run things. At that point, after just about 15 or so minutes of interaction, the time would have been yours to do the very first module, which is the nervous system's embryology. So I'm optimistic folks have done that, and you've come today, being Wednesday the 6th now for this hour. We'll spend some time as much as needed to review that material, a combination of both the general embryology of the system, which includes neurulation primarily, and then, of course, the section that was a little bit shorter dealing with neural crest cells, their derivation and eventual derivatives themselves. Next, of course, today, the balance of your morning, as much time as you have, I'd like to encourage you to go through the histology portion of the block, and that would just be the second module right here. There's not as much material in the histology section. I'd like to spend a bit of time maybe foreshadowing some ideas that I'd like you to keep at the forefront as you go through that material later this morning. But before we do that, let's look at this first module, the embryology. Let me open the floor for just a moment first and ask folks to chime in if you have any comments, any thoughts, any ideas, as there may be more fresh now after having just been gone through that material. What do you guys think? Cool, uncool, interesting, not very?
Pretty cool. I have a question for you, Dr. Reynolds.
Say again?
I said it was pretty cool. I have a question for you, though. Can you hear me?
I can, yes.
Just making sure my computer's working. Where are the eyes derived from? I couldn't tell if it was the, let's see here. if it was the diencephalon or the mesencephalon.
Yes, the former. It's from those are both from the diencephalon.
Okay.
So what I'd like to do is walk through the general idea of forming, first off, the neural tube. Got to do these in order, right? The neural tube, and then the next step is the formation of the 3-vesicle brain. You need to know the terms for those. And then the next step would be the 5-vesicle brain. And then at that point, you could spend some time looking at where things derive, right? So one idea, Jacob, you might be thinking of is a slide like this, where it has each of the 5 vesicles, and of course the spinal cord, which is not considered a vesicle, in red, and then in black, some of the highlights on what they become. And there are the eyes. They are part of the diencephalic derivatives, if you will. So let's look at that together. And then if the video is kind of still somewhat fresh for some of us, I did go through and look at some, I would say, beyond the scope detail. Some people might like it, and that's why it's there. But I'll go through and look at some of these specific organs. You'll recall I had several slides around slide 20 to 25 or so that gave you some additional details there. And that, again, is beyond maybe what the basal expectation is for this module, and I'll be sure to talk about that before we wrap up today. Thank
Question?
you, yeah, go for it.
So maybe you did just answer it, but I'm still a little confused. There's one slide that said in the mesencephalon, there's an anterior visual portion. So does that have anything to do with the eyes, or is that just knowing what you're seeing? Or?
That's correct. It doesn't have anything to do with the eyes. It's really the processing of information that comes from the eye. So the eyes, while they derive from the diencephalon, there is some visual processing matter that does come from the mesencephalon as well.
Okay, got it. Thank you.
You got it. Any other thoughts, knee jerks, quick responses to this idea? I mean, usually when we do this in a lecture setting, it's a bit more, it's different, right? We could detect each other's body language, interest level, and decide how deep to go on the fly. It's a challenge to do it like this, but we're up for the challenge. A lot of this stuff is probably the first time most, if not all of you, have heard of this material. So that's why I was kind of curious to see how it was received generally. Okay, received very well.
Great.
Thank you very much.
It was great. It was great. Thank you.
All right, let's look at some questions from the breakouts. There were 2 breakout opportunities. You have the breakouts at the very beginning, despite, I wouldn't say pressure, but requests. Some folks like to just look at the breakouts in real time with the answers, and that's cool because the breakouts are really well written, and who doesn't like to look at really cool breakouts? I get you. But my objective, and I'll be firm on this, is that I'd like you to look at the breakouts and be familiar with the materials by thinking them through on yourself, even struggling for just moments. And then note in the back of your mind, you will get the keys. Today I posted them really early, just the day after you would have otherwise got them yourself. So they're in there. Today's key I'll post a little bit later as well, the one for the histology, but they're both in there. So you're welcome to peek at those. Now, if you'd like, we'll look at them together right now. But again, I still would like you guys to go through and at some level, battle through individually or in small groups with the details that you can come up with on your own. So the first one, thought I'd do something different and show a fairly recent news article, publication that deals with this molecule known as neurotrophin-3. For those of you that are taking the time to be a part of the group, we're now up to 34. Welcome aboard, everyone. I'll go ahead and divulge the fact that neurotrophin-3 or NT3 is something that's interesting, but will never appear on any quiz or assessment. I won't ask any details about this particular research article, but I would have hoped you would have at least read through these dozen or so lines of material and thought about it and thought, wow, that is kind of cool. And it does reinforce maybe what I know about neurite outgrowth Anyone want to maybe start, as opposed to me maybe reading a few salient lines? Anyone want to start and just maybe provide some knee jerks or thoughts about what you picked up on from reading this abstract?
So I guess this is for wound healing, but also like development in utero, right? It's kind of the similar process.
Yes, for sure. There's some embryologic basis, so things that are pre-programmed in utero for sure. But this particular research group is focused on wound healing, evidently, in the skin. Notice they published it in the journal Pain. So it does give away a little bit about where their specific focus is. But it, in a general sense, talks about neurons, right? And neurite outgrowth. Who could define what neurite outgrowth is really quickly? Please.
Isn't it? It's just when I can't remember the name of them. The is it a podocyte? Anyway, there's a cell that goes and kind of explores and sees whether the environment is conducive for the nerves to live in. And
Perfect. Excellent.
Kind of take a home.
So if you would allow me some liberty, Stephen, podocytes specifically are going to be in the kidney, but they do have little feet extensions. You got it. So neurons will grow in a process known as neurite outgrowth. They're just simple cells initially, and if you imagine a cell in your mind, you just got a big spherical thing with a nucleus in the middle. They don't look like the neurons that you're familiar with long axons, right? Axons in you could be about 1 meter long, all the way down to your big toe from your inferior portions of the spinal cord. So that cell is much different than kind of a spherical looking cell. So a beginning cell that's more spherical will undergo a process of neurite outgrowth. It happens in utero, happens during development, very rarely happens postnatally, but some evidence suggests that you might get a few neurites postnatally with injury. Regardless, you have extensions. They're called growth cones that go out and they sample the local environment and then allow for branching to occur and growth. Those branches will derive dendrites or axons, depending on the direction of their growth. And each of those growth cones have little foot-like extensions called filopodia, which is what Stephen was thinking of. So let's look at this thing right here. Neurotropin 3, NT3, is a target-derived neurotrophic factor. So what that means is it's derived by the ultimate target. Let's say you're a neuron hanging out in your spinal cord, and you need to go out and innervate some muscle in your big toe. Everybody wiggle your big toe for a moment. See, you've done it. You've neurite outgrowth to allow that to happen, and you should be appreciative of that, right? So this is a target derived. Let's imagine if we use this loose example, this is derived by the musculature in the big toe, and it's a neurotrophic factor. It feeds and nourishes and supports the growth of neurons. And in this case, it'll regulate the sensory neuronal survival and growth, okay? So that is basically a quick introduction to that. Let's transition from the big toe idea now because we have new information in the second sentence. We report that NT3 plays a critical permissive role. So it releases signals that permit new things to happen in cutaneous skin, sensory nerve sprouting, and that contributes to pain and sensitivity following wound healing. So you can take young animals, model organisms, and induce in an approved, humane way, pain and sensitivity in their skin. And you would assume, therefore, that NT3 would have been one of the molecules, one of the chemicals that was necessary in allowing nervous system cells to sprout or neurite outgrow to that area. And I won't go through the rest of it in as much detail, but the next several sentences kind of summarize some of the work that they've done. The fact that if you block NT3, you don't get that response, further proving that it has a role in neurite outgrowth, etc. But at the end here, they conclude that if you upregulate NT3 in skin that's been wounded, it'll mediate the sensory nerve sprouting and increase hypersensitivity to pain following injury. So is this a modality for neuronal care in areas that are destined to be wounded or areas that are maybe wounded neonatally. We don't know just yet, and follow-up publications may suggest that. But regardless, it provides some of the first evidence. I know it looks like it is about a decade old, the work here, but it provided some of the first evidence of a specific molecule that has a role as cells undergo neurite outgrowth. Let me see if I can find that picture that you're likely familiar with from the presentation. Here it is. This is where axons and dendrites extend by way of growth cones that have philopodia. And let me see if I can mute this. Here's the video that took the narration off, but these are just images that show individual neurons like this one right here that are reaching out chemical sensing extensions, these growth cones, and saying, hey, I like this neuron over here, and let's form a connection, a circuit, as it were. All of this is happening billions and billions and billions of times over in an embryonic and fetal brain as connections are made. And that was the hope for that particular exercise. Do I know what neurite outgrowth is? And then some specifics there. So hopefully that was helpful. All right, the next section of this breakout asks you to describe the process of neural tube closure. You're welcome to read through that red text blurb a little faster than I could probably, but it basically summarizes the main concepts behind neural tube formation. You might remember images like this one. I know it's a little bit small. Let me make it a little bigger. That show the overlying surface ectoderm being induced by the notochord. These cells will then thicken, get larger, depress inward. That's the median hinge point of the neural groove. These two green sections are the lateral hinge points, and they'll zipper together right there and attach. At the point of fusion, these crest cells will delaminate, I talk about right there. That means they separate from the lamina or the layer of cells and become individual migratory cells. Those will do neural crest cell things later. But here's a really nice now fused neural tube right here along the midline. Similar to maybe this slide right there. Fusion happened right there. And then there would be zippering superiorly and inferiorly, forming this very nice tube of nervous tissue. All of that nervous tissue is derived from the ectoderm. Next, what we'll do is we'll form it into the 3 and then the 5 vesicle brain, and then it looks like the brain you're familiar with in a postnatal sense. I mentioned here at the end some ideas about the genetics, and if you'll recall from the presentation, I go through some of the details. A lot of these genes are critical, but the ones I'd like to focus on would basically be the FGF8 in sonic hedgehog. This is the anterior neural ridge, the first part of the brain. See the big bulge, prosencephalon right here. But all of this is controlled by sonic hedgehog development along the midline, and then a series of FGFs. So later on, if you could recognize FGF and ANR and second or sonic hedgehog, that would be more than enough for what I believe you need to know. Who has a follow-up question to this second question here? Everybody okay with that? Any extra detail needed or requested? Okay, so while I decided not to go through as much detail this year for the D3s. If you have defects along the line of any of the neural tube closure areas, there could be a series of birth defects. So I did not talk about spina bifida. That's more in the inferior part, lower part of the back usually. That's where the tube fails to close down here in the caudal region. Failure to close up here, again, is another type of defect that I'm not asking you to know this time. That's called anencephaly, where you don't have a brain really that forms up here near the top. So there are many different types of neural tube defects that are possible. If you remember, we did go through that when we described embryology generally 2+ years ago when you first came to campus, and we won't do that again this time. if that's all right. That's how we'll do it. Any questions about neural tube closure? If I can be of assistance, I'm happy to do so.
Question for you about the healing after hydroencephaly.
Uh-huh.
Patients that have that from birth, it said that there were those 2 treatments that you can do.
Yeah.
Is their skull then going to refuse? Are the bones going to come back together after they get that excess fluid out?
There is some indication that that's possible, but it's all about the timing. So the sooner this is detected, the better, and the shunt to remove the fluid, the better. But there is seemingly, although it's a gray area, point of no return where cranial defects are irreversible. So that was on a question where down here that we'll look at a little bit. Did you pick up anything else from like a search? What can you help us with, Stephen, on that?
I was just looking online a little bit to see. There were a couple of videos of people that had talked about like living with hydroencephaly, but it was, I don't know, I didn't get real conclusive evidence of whether they were totally A-okay or if they still had some serious side effects and whatnot afterwards.
Yeah, there are some neurological deficits, mental retardation, and those types of things is understandable. But if you're looking at images like this baby right here, a lot of this is going to be irreversible. It's really a function of timing. As soon as you could shunt that fluid out of the way to where there's no long-term effects, the better.
Awesome. Thank you.
You got it. Okay, if there are no other questions about the neural tube closure stuff, pull this out of the way. Let's look at this one. Provide context for the primitive brain with 3 vesicles and then the later brain with 5 vesicles, and various flexures are mentioned in there. So this was an opportunity for you to essentially summarize what the 3 and 5 vesicle brain looks like and what the names are. You'll remember from the video that I'd like you to know the names of those. So if you were to come up with a mnemonic or just reinforce it in a repetitive fashion, going over the terms, either way would work. But there are a lot of genes, again, FGFs and sonic hedgehog I'd like you to keep at the forefront, but many genes work together to remodel this neural tube after it fuses. The undifferentiated, just continuous tube will bulge and segment and transition quite extensively. The 3 vesicles you'll want to remember by name in order would be the prosencephalon, mesencephalon, and rhombencephalon. The primitive brain includes 2 different flexures. You'll recall the cephalic and the cervical flexures are the 2 flexures that are associated with the 3 vesicle brain. Let's see if I can find that slide here. This is the image that we're considering, where it's the prosencephalon, once again, mesencephalon and rhombencephalon. This is the cephalic flexure right in the middle of the mesencephalon. And there is the cervical, right where the neck is, flexure right down there. So continuing on, the 5 vesicle brain will next occur following additional segmentation. The telencephalon and diencephalon come from that first segment, the prosencephalon. Mesencephalon remains unaltered, remains the same in the 5 vesicle brain. And then in alphabetical order, metin and myelin, the 2 M ones for the last part of the rhombencephalon would then emerge in that sequence. Now, while there are 5 vesicles that appear, more detailed embryology would suggest additional segmentations, right? I did mention how the rhombencephalon is highly segmented compared to other areas. And even further still from the 5 vesicle brain, there's some more segments. We won't look at those, but I would like you to know the 3 and then the 5 with their names in order. And then some flexures. An additional flexure at the 5 vesicle brain, the pontine flexure, also emerges. And it, see if I can show you a picture of that one. Here's an image of the 5 vesicled brain. Telen, dien, mesen remains the same, metin and myelin down here. And in between those 2, right in the rhombomerric flexures, or segments rather, would be this pontine flexure, that 5th one there.
I think in your video.
Say again?
In your video, did you say that the cephalic separates the brain from the spinal cord. Is that correct? Like that?
I would say the cervical one would right here.
Okay. So I might have got those confused in my notes. I was going to fix that.
Yeah, the cephalic one is right up here. It will separate maybe the majority of the brain. All this right here will be the cerebrum. You know, if you were to imagine this right here, that's a huge portion of what the final brain will look like, the cerebrum up here. And you could say that's separated from the rest. This is the brain stem, the pons and the medulla oblongata. Those two are separated by way of that flexure, the cephalic flexure.
Okay.
But
and then where did you say the, oh, sorry.
Go ahead.
Where did you say the neck would be? So is this the cervical flexure, is that where the neck would be?
Yep. So here's the cervical flexure. Here's the spinal cord, comes right up to the base of the brain, does not include the brain stem, pons, medulla oblongata, etc.
Thank you.
You got it. Now, in order for all of these events to occur, and this maybe provides some cellular context, there's a unique cell proliferation program. So proliferation is going like crazy. And by the way, all of these cells in your brains now have no mitosis at all, no proliferation, zero. So what you have is what you have, and you need to take care of them. But the cellularity that's necessary for this huge, massive growth, which might have been appreciated a bit when you looked at this video here, all these different things that need to happen, the formation of the various vesicles and the extensiveness of this very simple tube becoming a robust brain, will be driven by the proliferation of the cells involved. So that's what these next couple of slides here suggest. You'll remember that there was this external limiting membrane, and then on the inside, the luminal side is where the actual mitosis happens. But it's pretty unique because the placement of the cells here will kind of vary as it's pseudostratified, goes through proliferation, and then they'll delaminate, really disconnect from this luminal portion right here and become lots of different cell types. So I thought that was unique and fun and even worth spending a minute talking about. So I'm hopeful you did as well. Any questions on that one, the 3 and 5 vesicled brain, before we transition to move to the next? Everybody okay? Please interrupt as we go. You don't have to wait for my requests for questions. Interrupt as we move along. Asks you next to highlight neuron formation, talking about neurite outgrowth and stabilization. We started today talking about neurite outgrowth a bit, but this summarizes what we've considered. Hopefully now you're appreciating the fact that neurons will differentiate from the various primitive neural cells in the neural tube. They'll receive chemical messages to induce outgrowth of neurites, growth cones, filopodia, all those terms that you're familiar with. These will become axons and dendrites. But over the process of forming connections with neighboring cells, those neurons will stabilize connections. And that's basically the process of reconfirming or reinforcing a sound connection between neurons that should exist in a circuit, whether it be a simple reflex or integration of information that you receive from the eyes or ears, etc. Good sound connections are maintained and stabilized. Others that are too weak, unutilized, not necessary, or problematic, maybe they don't line up just as well as they should, those will be weaned out by apoptosis. And there were a few little videos, I think, if I recall, at the end that kind of talk about this idea of stabilization of signal. All right, the next one question asks you to trace specific organs to their origins within the 5 vesicled brain. And there were several slides that were redundant. Gave you a chance to do that. Let me go to the beginning and find maybe one of the first few. Like this one here is one that shows you the telencephalon and diencephalon, both from prosencephalon, and there are specific derivatives in pink text for each. The mesencephalon is this one right up here. And then the metin and myelencephalon is also shown with pink text, specific derivatives. And you could use this type of a slide here after initially doing it on your own with no crutch. Or you can use the one that looks like this. It's very similar, although it might be easier for some of us to do that in kind of a list or tabular type form. Regardless, either of those two would work. And I have a tendency to ask questions like this, talk about fully formed neural structures and where they come from embryologically speaking. So this would be a good exercise for you to walk through a few times maybe. All right, now we're going to end up with the last two questions. I think Stephen had a question about the hydrocephalus idea. You were asked to look at the significance of it, maybe paraphrase it, even do a search and look for some treatments. These are some of the main ideas that were from the presentation at a minimum. Are there any ideas that you'd like to make known regarding hydrocephalus from either your own research, online search, etc., experience, whatnot? Is what I wrote sensical? Is there any areas of that blurb there that need clarification or augmentation in any way? So in the presentation, we first off talk about the mesencephalon. That's a portion of the brain. You can see it's mostly white matter, specific tracts, etc. I think there's a picture behind that baby. There it is. So it has a really large neural canal early on, and it should, by way of growth of the tissue, be kind of made smaller, if you will. But that central aqueduct, whenever it becomes blocked or occluded in any way, this hydrocephalus would result, relying or returning rather images that look like this. This is probably a less severe case, and the baby down here looks like she's from area 51 is a more severe case. But these are some of the details down here. The cortex would start to thin, and therefore you'd have damage and anomalous growth of the cerebrum, and therefore deficits of neurological function. And then there was some discussion about the shunts, and these were the two that I talked about in the video. But there are other opportunities for treatment as well. Anyone want to add anything to what you found online or elsewhere? Or is this, do we feel sufficient for both defining what hydrocephalus is and some possible treatment?
I mean, I just looked up VP shunt, VA shunts, just to see what that even stood for. It's ventriculoperitoneal. So right, going into the peritoneum, just releasing that fluid. Does it just get like cycled into lymph nodes and resorbed?
Exactly. The name of the game is just get the fluid out of the brain. Let's dump it off somewhere else, either directly into the circulation, right, for the VA shunt, or right into the peritoneal cavity. It can be reabsorbed either lymphatically or otherwise. And, you know, the key is just to get it out of the brain and relieve that pressure. Brain tissue, lung tissue, a lot of the tissue that we'll talk about embryologically speaking is sensitive to physical hindrance. So if there's too much fluid in the way, it's going to trade off and limit the amount of growth for the tissue and therefore lead to long-term deficit. So we can get it right down here into the peritoneum. We can send it right in here into the atrium. Both of these have been shown experimentally and clinically to be effective. So those are probably the key areas there that are done. All right, thank you everyone for participating there. Let's look at the last one. One more hammer home of the idea behind neurite outgrowth and stabilization. Both are related, right? You got to outgrow some neurites in order to make some connections, and those that you choose are most helpful to allow you to respond in reflex ways, or to walk, or to suckle, whatever behavior you need, those will be stabilized. You need those things to survive. So stabilization is a subsequent process after neurite outgrowth that will reinforce connections. Okay. And hopefully there's some degree of permanence behind those connections. Once you learn to ride a bike, you never forget, right? Isn't that something? All right. Any thoughts or overall concerns that you have that I can be of assistance in clarifying? Let's take the stage there. Okay, so let me come back to the PowerPoint for just a moment before we look at the second breakout. Breakout number 2 is this one here. But I'm going to race through some ideas. We only have 50 slides in this one, but the central nervous system involves a discussion first off about neural tube closure with some of the genetics. I'm hopeful that most, if not all, are comfortable with that at this point. The neural tube then will differentiate into 3 vesicles, pardon me, and then the 5-vesicled brain, and we've been through that a few times, and I'm hopeful that's familiar enough to all. There were some opportunities for you to look at adult or maybe postnatal derivatives of those various vesicles, and there should be some understanding there. Proliferation and neurite outgrowth. And then there was this idea of talking about some of the specific sections in the brain. Here it is right here. So at this point, you'll remember from the lecture recording that this is some extra material. If you have the bandwidth to memorize what happens in the diencephalon in the 5th, 6th, and 7th week, go for it. That's some good trivial information, but it's beyond the scope. I'd like you to know the basics. Okay, so this is some detail that gives you some additional thought about what the diencephalon becomes. If you knew from right up here that the diencephalon becomes a thalamus, hypothalamus, pituitary, and eyes, I'll add in the eyes from this one here. I think that's great. But if you wanted to know a bit more of the timing and the developmental changes that occur, these slides are helpful for that. Here's the pituitary gland. It's an interesting scenario that you will need to know about, but not just yet. Let's hold off on that until we look at the endocrine section of this block, the second half after your assessment here. I'll be back to talk about the infundibulum and Rathke's pouch. It's very helpful in a dental setting, developmentally speaking, and we'll talk about this for the pituitary gland. At this point, know it right here. Pituitary gland forms from the diencephalon. I feel good about that. But there is some detail here. If you wanted to jump ahead and close some of the loops, you're welcome to do so. The mesencephalon is a situation where we talked about hydrocephalus and the cerebral aqueduct. There's not much else going on there. It's mostly a relay center, a bridge point, if you will, between the higher centers of the brain, cerebrum, for example, and the brain stem. So that's what we looked at for the mesencephalon. And then down toward the brainstem area, we looked at the pons, the cerebellum, and the medulla oblongata. So all of this, these 3 or so slides, I didn't show this slide here, so don't be cranky if you see this for the first time, you don't have it. We looked at this one though, the cerebellum, the pons, and the medulla oblongata. Again, extra detail if you'd like to add to the overall understanding you already have regarding where those things come from, metin or myelencephalon. Is that okay with everybody? Anyone want to ask clarification on that last blurb I gave regarding the context of these 5 or 6 slides in the middle portion of your PowerPoint? Everybody okay? All right. Now, if I know you in the group here are interested in walking through the recorded lectures, looking at the PowerPoints, and then coming to these synchronous interactions. Not everybody does that. So you'll see here that in any of my PowerPoints, I will not write down every single thing that I say. I will not say everything that I write, but all of this gives you the overall feel for what my expectations are regarding the PowerPoint. So Glad you're here and you're able to get the clarifications you may have come seeking. All right, next in this blurb here would have been a transition from the CNS, developmentally speaking, toward the PNS, peripheral nervous system. Peripheral nerves grow in various fashion, and there's some transition about neural crest cell discussion. So we're going to look at that next in this next section of the PowerPoint, but here's a preemptive discussion about neural crest cells, where they come from and where they go, including parts of the digestive system's enteric control. So Hirschsprung's disease we looked at here, neurite outgrowth, nerve growth, etc., stabilization, all of that kind of finalize this first part of the PowerPoint. And I'm hopeful you're very familiar and comfortable with those general concepts. Let's pause there again and finalize group one by opening up the floor. Any questions or clarification you guys would like from that material so far? I feel you guys are doing well. It is early in the block though, right? So you're doing great. Keep up the good work. All right, let's look at the second half of the PowerPoint. Looks like it's only about a dozen or a baker's dozen of slides here. The neural crest. Neural crest forms from the two edges or boundaries of the neural groove. These are crests, really. And these green cells will then delaminate and become some cool things. So the first objective from the breakout was to draw a cross-sectional view of a closing neural tube and show where the crests are. I didn't do that, but I just stole this picture here. These are the crests on either side, and you may have decided to draw one out yourself. Regardless, it would have been this population of cells right there. Now, these cells will go through a series of steps in order to prepare for migration. Let's break the junctions between neighboring cells. Let's lose our polarity, etc. And then we're free at last. And we can, in a mesenchyme fashion, travel to some cool places. Here's the neural tube that's closed. Here's the notochord that orchestrated the process. But these cells right here from the crest are pinching off and they're moving. This is a somite, intermediate mesoderm lateral plate. So they're going to navigate around this thing and then just go to some cool places and become things. I spent some time talking about these two hypotheses. Do they come with loaded information, or do they have to wait till they get there to get some instructions? In fact, it's both. But then I looked at three different populations of neural crest cells. I would like you to be familiar with these three different groups of crest cells. So let's jump back into the breakout and make sure we've connected the dots. All right, first question here is, what do neural crest cells do in terms of normal nervous system development? Well, here's kind of a summary of where these cells have come from and some of the things that they become in a specific nervous system only effect. Skimming through this blurb right here, are there any details there that are quirky, hard to follow, or need clarification on? Okay. So a very general assumption about where they are located. Where they are located up here in the crests. When they fuse, they'll then, at the point of neural tube closure, they'll migrate based on the new environment that they migrate to. They'll differentiate into some additional structure, or they have some genetic pre-programming that they bring with them. And once they're established in their new locale, they'll become those derivatives. Both are documented to occur quite extensively in the literature. Specifics for the nervous system include cranial nerve ganglia. So the ganglia of the cranial nerves, 12 pairs of cranial nerves that Professor Novak will walk through with you, if not already in previous blocks. She's definitely going to do it again this time. Parasympathetic nerves in the gut. Remember those nerves and ganglia in the gut are problematic when Hirschsprung's disease is manifest. some sensory ganglia and sympathetic neurons and ganglia. All of those would be nervous system specific derivatives of neural crest cells. Someone unmuted and want to ask a question, or is that an inadvertent unmute? Any questions? Okay. A follow-up to that would have been some non-nervous system derivatives of neural crest cells. So I basically, there's an extensive list of structures. I'm not talking about all of them in this particular PowerPoint, but if you look at some of these populations here and then the table that I gave you, both those that have been mentioned and those listed are listed here in this response. Dermal bones of the skull. This is all from the cranial group right up here. Dermal bones from the skull, odontoblast, fragile arch. It should be pharyngeal arch, cartilage, facial dermis. All of that would be from the cranial group. The circumpharyngeal group of neural crest cells can make outflow tract and great vessels of the heart, thymus and parathyroid and thyroid. And then melanocytes is the most common derivative from the trunk neural crest cells. Any questions on some of that? Kind of follow up, there's some redundancies in this breakout as you've seen, but the three sources of neural crest cells again are these groups here. The basis for them leaving, the cranial group leaves before the neural tube closes, whereas the trunk group leaves after the neural tube closes. For the most part, in the cranial group, the origin, where it comes from, will pre-program its differentiation program, and that's not as indicated elsewhere to the degree that it is in the cranial group. But these are just some of the little caveats that I talked about when I described each of these three groups of neural crest cells. I didn't write them again, but some of the derivatives would have been some of these responses up here as well. Okay, our final question, unless there are any others, would be this one. Some abnormalities or anomalies that result from abnormal neural crest cell behavior. I tried to shy away from as much of the pathologies that may reemerge later on when Dr. Awan goes through that, but these are at least some of the ones that we spoke about. Obviously, if you're familiar with some of these derivatives of neural crest cells, when those derivatives fail to form, you would expect to see the diseases that are kind of summarized both in the breakout here and then as well in the PowerPoint that I gave you. All right, so let's open the floor to the group. Any thoughts about neural crest cells, where they come from, how they behave, where they go, or what they become? Okay. All right, let's do this then. As we, it looks like I had a few typos in this one. If you noticed as I was going, I corrected them. If you wanted to do that on you, well, I'll replace this one over here when we're done with our interaction. I'll do that in a moment. All right, so today for the remaining portion of your time, you have asynchronous time for you and your breakout groups, and you'll go over the nervous histology and nervous tissue materials. Here is, again, the document that we're reading together. Here's the PowerPoint that you're welcome to peruse in a static fashion. This is the video of me walking through the PowerPoint with you, and there's a singular breakout is all. So there's not as much material here that you're expected to know by mass, I guess, compared to other modules. So if you want to start working ahead after you get done with this, you're free to do so. But I did want to make sure that I provided a few thoughts for you at the outset. Here it is. So the next module that I have with you after histology is the physiology section. And I was trying to post it, but my file got corrupted, so I'm gonna have to re-record that. So I do apologize. It's not up yet. It'll be up there in a day or two. However, let me just mention that for this second PowerPoint that you're looking at today, the histology, there are only 37 slides, and there's not a ton of material. The basis of nervous system histology is to understand the neurons, as well as the glial cells. There are 6 supportive cells in the nervous system that I'd like you to be familiar with, and those are talked about in this PowerPoint as well. But if you've already been through this, or if not, if you're getting ready to do so, I'm hopeful that you'll see that I've started to incorporate a little bit of the physiology as well. So in this PowerPoint, you'll go through some of the main anatomical considerations of the cells, but then I do talk about this idea of resting potential, graded potentials, and action potentials. Action potentials right here, this little dude with the gun. So there is a kind of a preemption that allows you to kind of get your mind straight on some of the physiology as well. Some of this will obviously all of this will be reinforced in that next module that I have down here after Professor Novak, this physiology. I'll post that stuff in a day or so, like I say. But I just wanted to make sure that you're primed for that. There's going to be some histological and physiological aspects talked about in your presentation today. Okay, I think that's all that I really had come prepared with. I've come prepared with. So are there any thoughts or questions or any areas of confusion that might still remain from the embryology that I can be of assistance clarifying? Okay. All right, keep up the good work. Stay on top of things. Please feel free to reach out and ask questions. If you have them, email is easy. I get right back to folks that send me notes that way, so I'm happy to do so going forward. Let's meet again another time. You'll go through nervous and histology, and I won't meet with you again until the 15th. I'll drop an email once I've posted that physiology stuff. But on the 15th, that's the final day that I meet with you before your assessment. We will, for an hour, go through the histology. We may not take the entire time, but we'll walk through the block that you're doing today, nervous system histology and nervous materials, tissue materials. Then you'll get to go through the physiology stuff, and then I'd like to circle the wagons back again for this final hour, review any concerns you have for the physiology. I'll give you some good review material. Your assessment is coming up the following week. So I'll provide you some good review material and some other thoughts as I'm getting ready to write assessments myself in that era. So I'm happy to do that. I did want to maybe mention this really quick. There's a review day on the 26th. I don't know if you guys want to get together. We could do something like this for a half hour, or I can give you a quiz, or I can just give you a bunch of stuff this day or whatnot. Let's pocket this idea and think on it, and let me know what you'd like to do if you want to get together on a review day. All right, the floor is yours. Any questions or comments?
Thank you, Dr. Reynolds.
Hey, thank you. Thanks for being here. Okay, looks like that's where we'll end. Thanks, everybody. Appreciate it. You guys take care, and we'll see you again real soon.
Thank you.
Bye now.